Prophase, which is the phase. This is the most dramatic period. This is the most dramatic period of the cell cycle, involving a major recognition of virtually recognition. This is the most dramatic period of the cell cycle, involving a major recognition of virtually all to phase G2 phase of interphase. In the SN2 phase, the new DNA. In the SNG2 phase, the in the SNG2 phase, the new DNA molecules formed are not distinct but intertwined. Prophase is marked by the initiation of condensation. Prophase is marked by the Prophase is marked by the initiation of condensation of chromosomal material. The chromosomal material becomes untangled during the process of chromatin condensation. The centromere which had undergo duplication during S phase, the centromere which had undergone duplication during S phase of interphase now begin to move towards opposite poles of the cells. The completion of prophase can thus be marked by the following characteristics event. Prophase, which is the first stage of karyokinesis. Prophase which is the first stage of karyokinesis. Prophase, which is the first stage of karyokinesis of mitosis follows the SNG2 phase of the interphase. Prophase, which is the first stage of karyokinesis. Prophase, which is the first stage of karyokinesis of mitosis follows the SNG2 phase of interphase. Prophase, which is the first stage of karyokinesis of mitosis follows the SNG2 phase of interphase. In the SNG2 phase, the new DNA molecule formed are not distinct but intertwined. Prophase is marked by the initiation of condensation of chromosome. Yes. Hello. It's cell cycle and cell division. Yes. Okay. At the onset of anaphase, each chromosome arranged at the metaphase plate is split simultaneously, and the two daughter chromatids, now referred to as daughter chromosome of the future daughter nuclei, begin their migration towards the opposite poles. As each chromosome moves away from equatorial plate, the centromere of each chromosome remain detached towards the pole and hence at the leading edge, with the arm of the with uh, chromosome remains detached from the pole and hence at the leading edge. Chromosome poles ends the leading edge with the arm of the chromosome trailing behind. Thus, anaphase stage is characterized by the following key events: centromere splits and chromatid separate. Chromatid moves to opposite pole. Chromatid moves to opposite poles. Telophase at the beginning of the final stage. Telophase at the beginning. Telophase at the beginning of the final stage. Telophase at the beginning of the final stage of karyokinesis. That is telophase. The chromosome that have reached the respective poles decondense and lose their individuality. The individual chromosome can no longer be seen in each of the chromatin material tend to collect at each of two poles. This is the stage which shows the following key events: chromosomes cluster at opposite spindle pole and their identity is lost and discrete elements. Nuclear envelope develops around the chromosome cluster. Nuclear envelope develops around the chromosome cluster at each pole, forming two daughter nuclei: nucleolus, Golgi complex, and ER reform. Nucleolus, Golgi complex, and ER reform. Cytokinesis. Mitosis accomplishes not only the segregation of duplication chromosome into daughter nuclei, karyokinesis, but the cell itself is divided into two daughter cells by the separation of cytoplasm, called cytokinesis. Cytokinesis at the end of which cell division gets completed, and at the end of which cell division gets completed in an animal cell. This is achieved by the appearance of furrow in the plasma membrane. Appearance of furrow in the plasma membrane. The furrow gradually deepens and ultimately join in the center, divide the cell cytoplasm into two. Plant cells, however, the enclosed by a relatively extensible cell wall. Therefore, they undergo cytokinesis by a different mechanism. In plant cells, wall formation start in the center of the cell and grow upward to meet the existing lateral walls. The formation of the new cell wall begin with the formation of a simple precursors. The formation of the new cell wall begin with the formation of a simple precursor called the cell plate that represent the middle lamella between the wall of the two adjacent cells. At the time of cytoplasmic division, organelle like mitochondria and plastid get distributed into at the time of cytoplasmic division, organelles like mitochondria and plastids get distributed between the two daughter cells. In some organisms, karyokinesis is not followed by the cytokinesis. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinuclear condition arises, leading to the formation of synthesis liquid endosperm in coconut. Significance of mitosis. Mitosis or the equational division is usually restricted to the diploid cells only. Uh, Mitosis or the equational division. Mitosis or the equational division. Mitosis or the equational division is usually restricted to the diploid cells only. In some lower plants and in some social insects, haploid cells also divide. However, in some lower plants and in some social insects, haploid cells also divide by mitosis. It is very essential to understand the significance of this division in the life of an organism. Are you aware of some examples where you have studied about haploid and diploid insects? Mitosis usually result in the production of diploid daughter cell with identical genetic complement. The growth of multicellular the growth of multicellular organism the growth of multicellular organism is due to mitosis. Cell growth results in disturbing the ratio between the nucleus and the cytoplasm. It therefore becomes essential for the cell to divide to restore the nucleus-cytoplasmic ratio. A very significant contribution of mitosis is cell repair. The cell of the upper layer of the epidermis, cell of the lining of the gut, and blood cells are being constantly replaced. Mitotic division in the meristematic tissue, the apical and the lateral cambium, result in continuous growth of plants throughout their life. Meiosis, the production of offspring by sexual reproduction includes the fusion of two gametes, each with a complete haploid set of chromosomes. Gametes are formed from specialized diploid cell. Gametes are formed from a specialized diploid cell. This specialized kind of cell division that reduces the chromosomal number by half result in the production of diploid daughter cell. This kind of division is called meiosis. Meiosis ensures the production of haploid phase in the life cycle by sexual reproducing organisms, where fertilization restores the Meiosis ensures the production of haploid phase in the life cycle of sexually reproducing organisms, where fertilization restores the diploid phase. We come across meiosis during gametogenesis in plants and animals. This leads to the formation of haploid gamete. The key features of meiosis are as follows. Meiosis involves sequential Meiosis involves two sequential cycle of nuclear and cell division called meiosis first and meiosis second, but only a single cell of DNA replication. Meiosis first and meiosis second, but only a single cell of DNA replication. Meiosis first is initiated after the parental chromosome have replicated to produce identical sister chromatid at the S phase. S phase से start हो, S phase के बाद से Meiosis first is initiated after the parental chromosomes have replicated to produce identical sister chromatid at the S phase. Meiosis involves prepare for homologous chromosome. Meiosis involves preparing for homologous chromosomes and recombination between non-sister chromatid of homologous chromosome. Four haploid cells are found at the end of meiosis second. Meiotic events can be grouped under the following phase: M1 and M2 phase. Prophase 1 from metaphase, anaphase 1. process. Prophase of the first meiotic division is typically longer and more complex. Prophase of first meiotic division. Prophase of the first meiotic division is typically longer and more complex when compared to prophase of mitosis. It has been further subdivided into the following 5 phases based on chromosomal behavior. Leptotin, zygotin, pachytin, diplotin, and diagenesis. Leptotin, zygotin, pachytin, diplotin, and diagenesis. During leptotin stage of the chromosome become gradually visible under the light microscope. The compaction of chromosome continues throughout leptotin. This is found by the second stage of prophase called zygotin. During this stage, chromosomes start pairing together, and this process of association is called synapses. Such paired chromosomes are called homologous chromosome. Homologous electron microscope of this stage indicates the chromosome synapses is accompanied by the formation of complex structure called the synaptonymal complex. This complex formed by the pair of synapses homologous chromosome is called the bivalent or tetrad. However, these are more clearly visible at the next stage. The first two stages of prophase first are relatively short-lived when compared to the next stage, that is the pachytin. The four chromatid of each bivalent chromosome during this stage the four chromatid of each bivalent chromosome become distinct and clearly appear as tetrad. This stage is characterized by the appearance of recombinant nodules, the site of which crossing over occurs between non-sister chromatid of the homologous chromosomes. Crossing over is the exchange of genetic material between two homologous chromosomes. Crossing over is also an enzyme-mediated process, and the enzyme involved in the recombinase. Crossing over leads to recombinase of genetic material on the two chromosomes. Recombination between homologous chromosomes is completed by the end of pachytin, leaving the chromosome linked at the sites of crossing over. This begins of diplotin. Diplotin is recognized by the dissolution of the synaptonymal complex and the tendency of the this beginning of diplotin is recognized by the dissolution of the synaptonymal complex and the tendency of the recombined homologous chromosome of the bivalent to separate from each other except at the site of crossing over. Dissolution with the chiasmata. These X-shaped structures are called chiasmata. In oocyte of some vertebrates, diplotin can last for months of year. The final stage of meiotic. The final stage of meiotic prophase first is diagenesis. This is marked by the terminalization of chiasmata. During this phase of chromosomes are fully condensed and meiotic spindle is assembled to prepare the homologous chromosome for separation. By the end of diagenesis, the nucleolus disappear and the nuclear envelope also breaks down. Diagenesis represents the transition of metaphase. The bivalent chromosome align on the equatorial plate. The microtubule form the opposite poles of the spindle attached to the kinetochore of homologous chromosome. Anaphase first, the homologous chromosome separate while sister chromatid remains associated at their centromere. Telophase first, the nuclear membrane and nucleolus reappear. Cytogenesis follows, and this is called the dyad of cell. Although in many cases the chromosome do undergo some disappear, they do not reach the extremely extended state of the interphase nucleus. The stage between The stage between the two meiotic division is called interkinase and the generally short-lived. There is no replication of DNA. There is no replication of DNA during the interkinases. Interkinase is followed by prophase second as a simple prophase, then prophase first. Meiosis second, prophase second. Meiosis second is initiated immediately after cytokinesis, usually before the chromosomes have fully elongated. In contrast, a meiosis first, meiosis second resemble a normal mitosis. The nuclear membrane disappear by the end of prophase. The chromosome again become compact. Metaphase second. At this stage, the chromosome align at the equatorial plate and the microtubule form opposite poles of the spindle get attached to the kinetochore of sister chromatid. Anaphase second. It begins with the simultaneously splitting. It begins with the simultaneous splitting of the centromere of each chromosome, which has holding the sister chromatid together, allowing them to move towards opposite pole of the cells. Shortening of microtubule attached to kinetochores. Telophase second, meiosis second and telophase second, in which the two group of chromosomes once again get enclosed by a nuclear envelope. Cytokinesis follows, resulting in the formation of tetrad of cells, that is, four haploid daughter cells. Significance of meiosis. Meiosis is the mechanism by which conversion of specific chromosome number of each species is achieved across generation is sexually reproducing organism. Even when through the process, parcel paradoxically result in reduction of chromosome number by half. It is also increase the genetic variability in the population of organisms from one generation to the next. Variation are very important for the process of evolution. When you look around, you see both living and non-living things. You must have wondered and asked yourself, what is it that makes an organism living, or what is it that an inanimate thing does not have which a living thing has? The answer to this is the presence of the basic unit of life, the cell, in all living organisms. All organisms are composed. All All organisms are composed of cells. Some are composed of single cell and are called unicellular organisms, while others, like us, composed of many cells, are called multicellular organisms. What is cell? Unicellular organisms are capable of independent existence and performing the essential function of life. Anything less than a complete structure of a cell does not ensure independent living. Hence, cell is the fundamental structural and functional unit of all living organisms. Anton von Leeuwenhoek first saw and described live cell. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. पहले सबसे पहले लिविंग सेल किसने एंटोन वॉन लेवेनहॉक ने देखी थी. एंटोन वॉन लेवेनहॉक. Anton von Leeuwenhoek. Hello, this is Anton von Leeuwenhoek. Anton von Leeuwenhoek first saw and described a live cell. Anton von Leeuwenhoek first saw and described a live cell. Robert Brown later discovered the nucleus. The invention of the microscope and the improvement leading to the electron microscope revealed all the structural detail of the cell. Cell theory. In 1838, Matthias Leiden, a German botanist, Matthias Leiden, a German botanist, examined a large number of plants and observed that all plants are composed of different kinds of cells, which forms the tissues of the plant at about the same time. Schwann, 1838, a German geologist studied different types of animal cells and reported that cells had a thin outer layer of which is today known as the plasma membrane. He also concluded, based on his studies on plant tissue, that the presence of cell. Schwann, German geologist, studied different types of animal cells and reported that cells had a thin outer layer, which is today known as the plasma membrane. He also concluded, based on these studies of the plasma tissue, that the presence of cell wall is a unique character of the plant cell. On the basis of this, Schwann proposed the hypothesis that the two bodies of animal and plants are composed of cells and produce of cells. Leiden and Schwann together formulate the cell theory. Leiden and Schwann together formulate the cell theory. This theory, however, did not explain as to how new cells were formed. Rudolf Virchow, 1855, first explained that the cells divided and new cells are formed from pre-existing cells. Omnia cella e cella. He modified the hypothesis of Leiden and Schwann to give the cell theory a final shape. The cell theory as understood today is all living organisms are composed of cell and product of cell. All cells arise from pre-existing cells. cell. An overview of cell. You have earlier observed in an onion peel and a human chick cell under the microscope. Let us recall their structure. The onion cell, which is a typical plant cell, has a distinct cell wall as its outer boundary, and just within it is the cell membrane. The cells of the human chick have an outer membrane as the delimiting structure of the cell. Inside each cell is a dense membrane-bound structure called nucleus. This nucleus contains the chromosome, who in turn contain the genetic material, DNA. Cells that have membrane-bound nuclei are called the eukaryotic, where cells that lack a membrane-bound nucleus are prokaryotic. In both prokaryotic and eukaryotic cells are semi-fluid matrix called cytoplasm accompanies the volume of the cell. The cytoplasm is the main arena of cellular activities in both the plant and animal cells. Whereas beside the basic nucleus, the eukaryotic cell have other membrane-bounded distinct structure called organelles, like the organelles. Ribosomes are non-membranous bound organelles. Ribosomes are non-membrane bound organelles found in all cells, both eukaryotic as well as prokaryotic. Ribosomes are non-membrane. Ribosomes are non-membrane bound organelles cell. Ribosomes are found not only in the cytoplasm. Ribosomes are found not only in the cytoplasm but also within the two organelles, chloroplast in plants and mitochondria and on ER. Animal cell contain another non-membrane. Animal cell contain also another non-membrane bound organelle called the centromere, which help in cell division. Cell different greatly in size, shape and activities. For example, mycoplasma, the smallest cell are only 0.3 micrometer in length. Mycoplasma, the smallest cell, are only 0.3 micrometer in length, while bacteria could be 3 to 5 micrometer. While bacteria could be 3 to 5 micrometer. The largest isolated single cell. The largest isolated single cell is the egg on ostrich. Among multicellular organisms, human red blood cells are about 7 micrometer in diameter. Lung cells are some of the longest cells. Cells also vary greatly in their shape. They may be disc-like, polygonal, columnar, cuboidal, thread-like, or even irregular. The shape of the cell may vary with the function they perform. Prokaryotic cell. The prokaryotic cells are represented. The prokaryotic cells are represented by bacteria, blue-green alga, mycoplasma, and PPLO. Prokaryotic cells are represented. The prokaryotic cells are represented by bacteria, blue-green alga, mycoplasma, and PPLO. Pneumonia-like organisms. They are generally smaller and multiply more rapidly than the eukaryotic cell. Eukaryotic cell. They may vary greatly in shape and size. The four basic shape of bacteria are bacillus, rod-like, coccus, spiral, vibrio, comma shape, and spirillum spiral. The organization of the prokaryotic cell. The organization of the prokaryotic cell is fundamentally similar, even though prokaryotes exhibit a wide variety of shape and functions. All prokaryotes have a cell wall surrounding the cell membrane, except in mycoplasma. Prokaryotes have a cell wall surrounding the cell membrane, except in mycoplasma. The fluid matrix filling the cell wall is cytoplasm. There is no well-defined nucleus. The genetic material is basically naked, not enveloped by nuclear membrane. In addition, in the genomic DNA, the single chromosome circular DNA. Many bacteria have small circular DNA outside the genomic DNA. This smaller DNA are called Many bacteria have small circular DNA outside the genomic DNA. The smaller DNA are called plasmid. The plasmid DNA conforms certain unique phenotype character to such bacteria. One such character is resistance to antibiotics. In higher classes, you will learn that the plasmid DNA is used to monitor bacterial transformation with foreign DNA. Nuclear membrane is found in eukaryotes. No organelle like the ones in eukaryotes are found in prokaryote cell except for ribosomes. अलावा कोई भी ऐसा नहीं जो उसमें पाया जाए. Prokaryotes have some for ribosome. Prokaryotes have something unique. In the form of inclusion, spatial different inclusions. mesosome in the lateral floor. They are essential in folding of the cell mesosome. Mesosome includes a non-membrane. Cell envelopes and its modification. Cell envelope and its modification. Cell envelope and its modification. Most prokaryotic cells, particularly the bacterial cell, have a chemically complex cell envelope. The cell envelope consists of a tightly bound three-layered structure, that is the outer glycocalyx, followed by the cell wall and the thin plasma membrane. Through each layer of the envelope perform distinct function. They act together as a single production unit. Bacteria can be classified into two groups on the basis of difference in the cell envelope and the manner in which they respond to the staining procedure during the gram phase. Through that they take up the chum stain for gram positive and the other do not, called gram negative. Glycocalyx differ in composition and thickness among different bacteria. Glycocalyx differ in composition and thickness among different bacteria. It could be loose sheath called the slime layer in some, while in other it may be thick and tough called the capsule. The cell wall determines the shape of the cell and provides strong structural support to prevent the bacterium from bursting or collapsing. The plasma membrane is selectively permeable in nature and intersects a plasma membrane is selectively permeable in nature and interacts with the outside world. This membrane is similar structure to the eukaryotes. A special membranous structure is the mesosome, which is form the extension of plasma membrane into the cell. These extensions are in the form of vesicle, tubular, and lamellae. This help in the cell wall formation, DNA replication, and distribution to daughter cells. They also DNA replication and distribution to daughter cells. They also formation, DNA replication, distribution to daughter cells. They also help in respiration secretion process to increase the surface area of the plasma membrane and genetic content. In some prokaryotes like cyanobacteria, there are other membrane extension into the cytoplasm called the chromatophores. Like cyanobacteria, chromatophore which contain pigment. Bacterial cell may be motile or non-motile. If motile, then they have the filamentous extension from their cell wall called the flagella. Bacteria show a range in the number of arrangement of flagella. Flagella is composed of three parts: filament, sulca, and basal body. The filament is the longest portion and extends from the cell. Longest portion and extend from the cell surface to the outside. Besides flagella, pila, and fimbria are also surface bacteria to the bacteria but do not play a role in motility. The pili are elongation tubular structure made of a special protein. The fimbria are small crystallized Ribosomal inclusion body. In prokaryote, ribosomes are associated with the plasma membrane. In prokaryotes, ribosomes are associated with the plasma membrane. Ribosomes are associated with the plasma membrane. In prokaryote, ribosomes are associated with the plasma membrane of the cell. They about 15 nanometer 20 nanometer in size, and they are made up of two subunits 50s 30s as which when present together at the 70s prokaryote ribosome. Ribosomes are the sites of protein synthesis. Several ribosomes may attach to the single mRNA and form a chain called polyribosome or polysome. Ribosome of a polysome translate the mRNA into protein. Inclusion body. Resolved material in prokaryote cell are stored in the cytoplasm in the form of inclusion bodies. These are not bound by any membrane system and live free in the cytoplasm. That phosphate granules, cyanophosphate granules, and glycogen granules. As vacuole are found in blue, green, and purple and green photosynthetic bacteria. Eukaryotic cells. The eukaryotes include all the protista. The eukaryotes include all the protist, plant, animals, and fungi. In eukaryotic cells, there is an extensive compartmentalization. Of cytoplasm through the presence of membrane-bound organelles. Eukaryotic cell possess a organized nucleus with the nuclear envelope. In addition, eukaryotic cells have a variety of complex locomotory and cytoskeleton structure. Their genetic material is organized into chromosomes. All eukaryotic cells are not identical. Plant and animal cells are different as the possess cell wall, plastids, and a large central vacuole which are absent in animal cell. On the other hand, animal cell have centrioles which are absent in almost all the plant cells. Let us now look at the individual cell and understand their structure and function. Cell membrane. The detailed structure of the membrane was studied only after the advance of the electron microscope in 1950s. Meanwhile, chemical studies on the cell membrane, especially on human red blood cells, enable the scientists to reduce the possible structure of plasma membrane. These studies show that the cell membrane is mainly composed of lipids and protein. Composed of lipids and protein. This is shown that the cell membrane is mainly composed of lipids and protein. The major lipids are phospholipids that are arranged in a bilayer. Also, the lipids are arranged within the membrane with the polar head towards the outer side, polar heads outer side, polar head towards the outer side, and the hydrophobic tail towards the inner part. This ensures that the non-polar tail is saturated. Non-polar tail is saturated. Hydrocarbon is protected from the aqueous environment. In addition to phospholipid, membrane also contains cholesterol. Later biochemical investigation clearly revealed that the cell membrane also possesses protein and carbohydrate. The ratio of protein and lipid varies considerably in different cell types. In human being, the membrane of the erythrocytes has approximately 52% of protein, 52% क्या protein, 52% protein and 40% क्या है 52% protein and 40% lipids. Depending on the ease of interaction depending on the ease of extraction, membrane protein can be classified integral and peripheral. Peripheral protein lie on the surface of membrane. Peripheral protein lie on the surface of the membrane, while the integral protein are partially or totally buried in the membrane. An improved model of the structure of the cell membrane was proposed by Singer and Nicholson. Singer and Nicholson 1972, widely accepted as fluid mosaic model. According to this, the quasi-fluid nature of the lipid enable lateral movement of protein within the overall bilayer. This ability to move enable lateral movement of protein within the overall bilayer. This ability to move within the membrane is measured as its fluidity. The fluid nature of the membrane is also important from the point of view of function like cell growth formation of intercellular junction, secretion, endocytosis, cell division, etc. One of the most important functions of the plasma membrane is the transport of the molecules across it. The membrane is selectively permeable. The membrane is selectively permeable to some molecules present on either side of it. Many molecules can move briefly across the membrane without any requirement of energy, and this is called the passive transport. Natural solutes may move across the membrane by the process of simple diffusion along the concentration gradient from higher concentration to the lower. Water may also move across this membrane from higher to lower concentration. Movement of water by diffusion is called osmosis. As the polar molecule cannot pass through the non-polar lipid bilayer. They require a carrier protein of the membrane to facilitate their transport across the membrane. A few ions or molecules are transported across the membrane against their concentration and from lower to the higher concentration. Such a transport as an enzyme-dependent process and ATP is utilized, and this is called active transport. Sodium and potassium pump. Cell wall. As you may recall, a living rigid structure called the cell wall. As you may recall, a non-living rigid structure called the cell wall. Form an outer covering for the plasma. Form an outer covering of plasma membrane of fungi and plants. Cell wall not only gives shape of the cell and protects the cell wall. Cell wall not only gives shape to the cell and protects the cell wall from mechanical damage and infection. It also helps in cell-to-cell interaction and provide barrier to undesirable macromolecules. Alga have cell wall made up of cellulose, galactans, vannans, and minerals like calcium carbonate, while in other plant it consists of cellulose, hemicellular, pectin, and protease. The cell wall of the young plant cell, the primary wall, is capable of growth, which gradually diminishes as the cell matures, and the secondary wall is found on the inner towards the membrane side of the cell. The middle lamella is a layer. The middle lamella is a layer mainly of calcium pectate. The middle layer, the middle lamella, the middle lamella is a layer mainly of calcium pectate which holds or glues the different neighboring cells together. The cell wall and middle lamella may be traversed by plasmodesmata which connect the cytoplasm of neighboring cells. Endomembrane system. While each of the membranous organelle is distinct in the structure terms of its structure and function, many of these are considered together as an endomembrane system because their function are coordinated. The endomembrane system includes endoplasmic reticulum, Golgi complex, lysosome, and vacuole. Since the function of the mitochondria, chloroplast, and peroxide are not coordinated with the above components, these are not considered as the endomembrane system. Endoplasmic reticulum, lysosome, endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. Endoplasmic reticulum, Golgi complex, lysosome, and vacuoles. Or function of the mitochondria, chloroplast, and peroxisome are not coordinated with the above component. These are not considered as a part of the endomembrane system. The endoplasmic reticulum. Electron microscope studies of eukaryotic cells reveal the presence of network of reticulum or tiny tubules scattered in the cytoplasm. That is called the endoplasmic reticulum, ER. Hence, ER divides the intracellular space with the two distinct compartments, the lumen inside ER and extralumen cytoplasm compartments. The ER often show ribosome attached to their outer surface. The endoplasmic reticulum bearing a ribosome on their surface is called the rough endoplasmic reticulum. In the absence of ribosome, they appear smooth and are called smooth endoplasmic reticulum. Rough endoplasmic reticulum is frequently observed in the cell actively involved in protein synthesis and secrete. They are extensive and continuous with the outer membrane of the nucleus. The smooth endoplasmic reticulum is major site for the synthesis of lipids. In human cells, lipids like steroidal hormones are synthesized the smooth endoplasmic reticulum. Golgi apparatus. Camillo Golgi first observed dense stained reticular structure near the nucleus. These were later named Golgi bodies. After him, they consist of many flat dish-shaped sac or cisternae of 0.5 micrometer to 1.0 micrometer diameter. These are stacked parallel to each other. Varied number of cisternae are present in Golgi complex. The Golgi cisternae are concentrically arranged. The Golgi cisternae are concentrically arranged near the nucleus with distinct convex cis or the forming phase and concave phase or the mature phase. Concave phase को mature phase कहते हैं. The cis and the trans phase of the organelle are entirely different, interconnected. The Golgi apparatus principally performing the function of the packed material to the delivered either to the extracellular target or secrete outside the cell. Material do not packed in the form of vesicle from the maturing phase. This explains why the Golgi complex remains closely associated with the endoplasmic reticulum. A number of proteins synthesized by ribosome on the endoplasmic reticulum are modified in the cisternae of the Golgi operator before they are released from the transphase. Golgi apparatus is the important site of formation of glycoprotein and glycolipids. Lysosome. These are lysosome. These are membrane-bound vesic- uh, vesicular structure formed by the process of packaging of in the Golgi apparatus. The isolated lysosome vesicle have been found to be very rich in almost all type of hydroxyl enzyme, hydrolase, lipase, protease, carbohydrates. Optimal active of the acidic pH. Optimally active at the acidic pH. These enzymes are capable of digesting carbohydrate, proteins, lipids, and nucleic acids. Nucleic acid vacuoles. The vacuole is the membrane bound. The vacuole is the membrane bound space. The vacuole is the membrane bound space. Remember, 90% of the volume of the cell. In plant, the tonoplast facilitates the transport of a number of ions and other material. In the plant, the tonoplast facilitates the transport of the number of ions and other material against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. In many cells, as in protists, food vacuole are found in engulfing the food particles. Vacuoles क्या है? The vacuole is the membrane-bound space found in the cytoplasm. It contains water, sap, excretory product, and other material not useful for the cells. The vacuole is bound by a single membrane called tonoplast. In plant cell of the vacuole can occupy up to 90% of the volume of the cell. In plants, the tonoplast facilitates the transport. In plants, the tonoplast facilitates the transport of a number of ions. In plants, the tonoplast facilitates the transport of a number of ions and other material against concentration gradient into the vacuole. Hence, their concentration is significantly higher in the vacuole than in the cytoplasm. In amoeba, the contractile vacuole is important for osmoregulation and excretion. Mitochondrion, unless spatially stained, are not easily visible under the microscope. Single mitochondrion, unless specifically stained, are not easily visible under the microscope. The number of mitochondria per cell is variable depending on the physiological of the cell. In terms of shape and size, also considerable degree of variability in observed. Typically, it is a sausage-shaped or cylindrical shape. A diameter of 0.2 to 1.0 micrometer, average 0.5 micrometer, and length 1.0 to 4.1 micrometer. Each mitochondria is double membrane bound structure with the outer membrane and the inner membrane divided in its lumen distinctly into its aqueous compartments. Outer compartment and the inner compartment. The inner compartment is filled with the dense homogeneous substance called the matrix. The outer membrane form The outer membrane forms the continuous limiting boundary of the organelle. The inner membrane forms the number of enfolding called the cisternae. Mitochondria में क्रिस्टी होते हैं. क्रिस्टी towards the matrix. The mitochondria में क्रिस्टी होते हैं. The cristae increase the surface area. Cristae increase the surface area. The two membranes have their own specific enzyme associated with the mitochondria function. Mitochondria are the sites of aerobic respiration. They produce cellular energy in the form of ATP. Hence, they are called powerhouse of the cell. The matrix also possesses single circular DNA and few RNA molecules, ribosome 78, and the components required for the synthesis of protein. The mitochondria divide by fission. Mitochondria divide by fission. Plastids. Plastids are found in all plant cells and in euglenoids. These are easily observed under the microscope as they are large. They bear some specific pigments. Plastids are found in all plant cells and euglenoids. These are easily observed under the microscopes as they are large. They bear some specific pigment, thus imparting specific color to the plants. Based on the type of pigment, plastids can be classified into chloroplast, chromoplast, leucoplast. Chloroplast, chromoplast, and leucoplast. The chloroplast contain chlorophyll. The chloroplast contain chlorophyll and carotenoid pigment, which are responsible for trapping light energy essential for photosynthesis. In the chromoplast, fat-soluble carotenoid. Chromoplast, fat-soluble carotenoid pigment like carotene, xanthophyll, and other are present. This gives the part of the plants an yellow, orange, or red color. The leucoplasts are the colorless plastids of various shape and size with stored nutrients. Amyloplasts store carbohydrate, starch. Potato Elaioplast stores oil and fats, where the aleuroplasts store proteins. Majority of the chloroplasts of the grain plants are found in the mesophyll cells of the leaves. These are the lens-shaped, oval, spherical, discoid, or even ribbon-like organelles. Have variable length 5 to 10 micrometer and width 2 to 4 micrometer. Their number vary from 1 parcel to chlamydomonas to 20 to 40 parcel of the mesophyll. Like mitochondria, the chloroplasts are also double membrane bound of the two. The inner chloroplast membrane is relatively less permeable. The space limited by the inner membrane of the chloroplast is called the stroma. A number of organized flattered membranous sacs called the thylakoid are present in the stroma. Thylakoid are arranged in stacks like the piles of coins called grana, similar granum, or the granular thylakoid. In addition, here the flat membranous tubules called the stroma lamella connecting the thylakoid of the different grana. The different of the thylakoid encloses a space called the lumen. The stroma of the chloroplast contains enzymes required for the synthesis of carbohydrate and protein. It also contains small double-stranded circular DNA molecule and ribosome. Chlorophyll pigments are present on the thylakoid.